Certain side chain conformations are energetically more favorable than the others. Any side chains longer than that of alanine can in principle have several different conformations. In A, you can see in ethane chemicals non-staggered and staggered, where you see the hydrogens just position at the center of formed by two atoms in neighboring carbons, avoiding just a possible steric hindrance between them, which is much different from the non-staggered structures where two hydrogens attached to the neighboring two carbons, they just position sterically in space, could overlap. So three possible staggered forms are produced, which are related by 120 rotation around carbon bond. These three conformations are not indistinguishable because simply all three positions are identical. However, in valine, as you see here, around these carbon-carbon bonds, two methyl group and one hydrogens. So this is the most stable among three possible staggered forms because bulky two methyl groups just position in smallest hydrogens compared to the other two structures, C and D. Methyl groups just position with bulky nitrogens. Okay, so side chains for the valine, B form is most stable conformation than the other. So this kind of stable conformation in side chains is called rotamer. Today, collections of these favored conformations for rotamer library are a standard tool in computer programs used for modeling protein structures. Of course, there are four non-covalent bonds contributing, making three-dimensional structures. Hydrogen bond, where oxygen or nitrogens, they just interact with the hydrogen that makes covalent bond with oxygen or nitrogen. Electronic interactions, positively charge it or negatively charge it. They just pull each other or push each other, depending on the charges. Hydrophobic interactions, non-polar molecules in hydrophilic compounds, they tend to together try to reduce the interacting surface with hydrophilic molecules. Van der Waals interactions. Any two atoms basically in close proximity tend to pull together. Even though the strengths of Van der Waals interactions are very tiny, ubiquitous among all sorts of atoms, could strengths as a result. Of course, there is a strong covalent bond between cysteine and cysteine, forming disulfide bond. This could increase dramatically protein stabilities. Most the secreted protein or localized on surface of proteins cells tend to have disulfide bond because of harsh environment. Side chains of 20 different amino acids have very different chemical properties. However, their chemical versatility is not unlimited. For this reason, other components like metals are frequently part of proteins. In this picture, iron to ions make bonds with negatively charged aspartic acid, glutamic acid, plus histidine. Even water molecules are also contributing, making stabilization. This is the part of enzyme ribonucleotide reductase contributing, making this kind of tyrosine free radical, which is involved in catalysis. In B, another metal, zinc, found in enzyme alcohol dehydrogenase. So in this case, 2 cysteine and 1 histidine binding to the zinc. Lastly, I just remind you types of proteins. In terms of composition, proteins could be either simple or conjugate proteins. For the conjugate proteins, depending on the conjugate, it could be lipoprotein, glycoprotein, metalloprotein. Okay, other parts are called cofactor. That could be either inorganic element or organic element. Organic element called coenzyme. Coenzyme could be utilized, could be synthesized from vitamin for many cases. Prosthetic group sometimes indicating conjugate when it is just being bound to the protein with a very tight affinity. Another classification might be depending on location and shape plus solubility. Non-membrane protein or membrane protein. Non-membrane protein may be either globular protein or fibrous protein. membrane protein might be integral or peripheral. That's it for this session.